Good day, we are the Group 4 and we'll be presenting about Plasmodium species, which are single-celled protozoan parasites. Moving on to their specific classification, they belong to the phylum Apicomplexa, class Aconioidacida, order Hemorosporida, family Plasmodiidae, and genus Plasmodium. So here the listed 5 species are particularly observed in the blood, which already signifies their main mode of transmission, which is through an insect vector. The Plasmodium species are responsible for causing malaria, which is the most significant tropical disease that affects humans, but generally vertebrates. It is preventable and curable. The organism is transmitted by the bite of the female Anopheles or Anopheline mosquito. For malarial infections, it is necessary to recognize the 4 primary erythrocytic stages of the organism as a means for identification. So these are the ring form, the trophozoite or growing form, the schisant or dividing form, and the gametocyte or sexual form. So first is the ring form, which is characterized by a blue cytoplasmic ring, which is supposedly connected to a red chromatin dot. Since the trophozoite is the growing form, the red chromatin dot has now increased in size and is surrounded with more abundant blue cytoplasm. The schisant, or dividing form, is characterized with divided red chromatin, and there is now the presence of brown pigment, which is referred to as the hemazoin or hematine, associated to products of RBC digestion. Gametocyte or sexual form, there's now the compact red chromatin mass and still the presence of brown pigment. There are 5 known species that cause human malaria, which are the Plasmodium vivax, Plasmodium ovale, Plasmodium malariae, Plasmodium falciparum, and Plasmodium knowlesi, even though P. knowlesi is usually causing zoonotic malaria. The Plasmodium vivax is the most common form worldwide, which causes benign tertian malaria, and may be morphologically indistinguishable from Plasmodium ovale.
And next, for its general morphology, the appearance of infected erythrocyte size is enlarged, then the presence of Scheffner's staplings is a finding associated with malaria, and it's important for species identification. So it is also present in all stages except for early ring forms. Its cytoplasm is irregular and amoeboid. So enteropocyte has a spread out appearance. The parasite pigment is golden brown, yet inconspicuous or not clearly visible. There are approximately 12 to 24 with an average of 16 merozoites that infect red blood cells, and all of its morphological stages are found in the blood. So here's the illustration of its signet ring form with the major characteristic component of a heavy red chromatin dot and blue cytoplasmic rings as shown here in the next slide. In the micrograph, a vacuole is present within the cytoplasmic ring. And next is the trophozoite form with the same characteristic component but has increased cytoplasm and amoeboid activity. And as shown in the next slide, there are small yellowish brown pigments, granules in the cytoplasm that increases as the parasite develops. So for the next, there are immature and mature schizont form. Basing from the illustration, there is now the absence of a chromatin dot and a cytoplasmic ring is both forms due to a developing modification. In the mature schizont, chromatin is divided and the cytoplasm shows varying degrees of separation into strands and particles. Pigments then collect in parts of the parasite as shown in the image on the left. The mature schizonts contain the 12 to 24 merozoites. Pigment collects in 1 to 2 clumps which are more condensed, and the parasite almost fills the enlarged RBCs as seen in the image on the right.
Last are the microgametocytes and macrogametocytes. Here, the previous chromatin bodies now become denser, forming a chromatin mass differing in the locations within a homogeneous cytoplasm. In the next slide, here are the micrographs. The left image shows the microgametocyte, which has light red to pink chromatin mass, which is diffuse and central, and it gives tint to the light blue cytoplasm. There is also the presence of yellowish-brown pigment throughout the cytoplasm with a size of a normal red blood cell. The right image shows the macrogametocyte, which has a small, compact, dark red eccentric chromatin mass with a dark blue cytoplasm. There are no vacuoles present, and abundant dark brown pigment is scattered throughout.
Plasmodium ovale. Plasmodium ovale infect immature RBCs or specifically reticulocytes. So a little history about this species. In 1900s, Craig described a malaria parasite in the blood of American soldiers returning from the Philippines that they had a peculiar morphological characteristics and a tertian fever pattern, and it is possible that he was describing infections with Plasmodium ovale. So after that, McPhee and Ingram in 1917 described also a parasite in the blood of a child in the Gold Coast that may also have been the Plasmodium ovale. So in 1922, Stephens published a full description of the forms in the blood and named the parasite Plasmodium ovale in recognition of the oval shape of some of the infected erythrocytes. And with that, Plasmodium ovale was the last of the malaria parasites of humans to be described. Next is its morphology. When you are infected with Plasmodium ovale, the appearance of erythrocytes or its size is enlarged and elongated, which is somewhat similar to Plasmodium vivax. Its stippling is present in all stages except in early ring forms, and the James dot is occasionally seen. The parasite cytoplasm is rounded, it has a compact trophozoites, and it is occasionally slightly amoeboid. The growing trophozoites have large chromatin mass. The appearance of its parasite pigment is dark brown and conspicuous, and it have a 6 to 14 number of merozoites, which have the average of 8, and all stages are found in circulating blood.
Plasmodium ovale ring form have sturdy cytoplasm and large chromatin ducts. Its trophozoites form have sturdy cytoplasm, large chromatin ducts, an oval form, and can be complex like irregular. Its cell border is commonly fibrated or ragged. Its Kiesen's form has 6 to 14 merozoites with large nuclei, clustered around the mass of dark brown pigment. On the other hand, the gametocyte's form is round oval and may almost fill the red blood cells. Pigment is brown with more coarse in comparison to Plasmodium vivax.
The third parasite that we will be discussing is the Plasmodium malariae. The morphology of the Plasmodium malariae are the following. The size of the erythrocyte is normal. This is because the parasite does not alter or cause any enlargement. The Schaffner's stippling that can be already seen is called Zeimann's dot. The cytoplasm of the parasite is rounded. The trophozoites is compact with dense cytoplasm. The pigment of the parasite is dark brown, coarse, and conspicuous. The number of merozoites is in the range of 6 to 12, with an average of 8 and occasionally arranged in a roseate pattern. Lastly, all stages of the parasite can be seen circulating the blood. Next, we will be discussing the microscopic features. The ring form of the parasite is similar to the Plasmodium vivax, but with blue cytoplasmic circle, and is smaller, thicker, and heavier. It also has a large chromatin dot. This is an example of the ring form of the Plasmodium malariae. The next form will be the trapezoid form. The trapezoid form has a compact cytoplasm with a large chromatin dot and has an occasional band forms or basket form with a coarse dark brown pigment that can be seen. These are the examples of the trapezoid form, the band forms, and the basket forms.
Now proceed to the next microscopic feature, which is the schizones. As you can see, there are two stages of schizones, the immature and the mature schizones. In the immature schizones, you can see here the cytoplasmic material and the chromatin bodies, while in the mature schizones, it has merozoites. Schizones of Plasmodium malariae have 6 to 12. Usually, it has also 8 to 10 merozoites, often arranged in a rosette or a regular cluster. Mature schizones nearly fill the normal size host of the RBC, pigment of course, and often peripheral. Schizones can be common in peripheral blood circulation. This is the appearance of the schizones on the microscope. So the Plasmodium malariae schizones have, like what I've mentioned, it has 6 to 12 merozoites with large nuclei clustered around a mass of coarse dark brown pigment. Merozoite can occasionally be arranged as a rosette pattern. So you can see here the schizones of P. malariae appearance in a rosette pattern. Next is the gametocytes. It has two types, which is the micro and macro gametocytes. The only difference between the two is that the macrogametocytes has a cytoplasm. They are both the same when it comes to chromatin mass and coarse pigment. And you can see the location of the chromatin mass. It is central in the microgametocytes, while in the macrogametocytes, it is located at the top. Plasmodium malariae gametocytes are round to oval with scattered brown pigment. They may also fill the infected red blood cell. So here is the appearance of the Plasmodium malariae gametocytes in the microscope. Gametocytes of P. malariae are compact and tend to fill the host RBC. There is no enlargement of the infected RBC, and sometimes there is a reduction in size. The cytoplasm stains blue and the chromatin pink to red. Abundant dark pigment may be scattered throughout the cytoplasm.
Our next parasite under the genus Plasmodium is Plasmodium falciparum. So falciparum is the species that causes black water fever and malignant tertian malaria, which is the most deadly form of malaria in untreated patients. So as discussed in our prelim lessons on malaria as to the erythrocyte affinity of the various species of Plasmodium, Plasmodium falciparum is known as a desperate parasite since all ages of RBCs may be infected, thus producing large amounts of toxic cellular debris and capillary plugs consisting of various blood elements. So for the morphology and cytoplasmic inclusions of the RBCs infected with Plasmodium falciparum, the appearance of infected RBCs appear to be normal in size and no significant distortions can be seen. Our falciparum can, however, infect multiple red blood cells, which is common for the parasite. For the Schaffner stiplings, the cytoplasm of the red blood cells infected with P. falciparum may contain Morris dots, which is the unique marker of your Plasmodium falciparum. So if we try to remember our mnemonics, Ferdinand Marcos, F is for falciparum and M is for your Morris dots. Next, the young rings of your Plasmodium falciparum are small in size, delicate, and often characterized with two chromatin dots. Your gametocytes of the significant species in the genus Plasmodium have a roundish to oval micro and macro gametocytes. However, for your Plasmodium falciparum, it has a crescent-shaped gametocyte, which is a distinguishing characteristic of your falciparum. For the appearance of your parasite pigments, It is usually black and appears to be coarse and conspicuous in your gametocytes. And the number of merozoites is usually 6 to 32 with an average of 20 to 24. Lastly, the ring form as well as the gametocytes can be seen in the circulating blood. So we move to our microscopic features. For our microscopic features, the typical small delicate ring form of your Plasmodium falciparum is similar with your Plasmodium vivax but consists of scanty cytoplasm connected to one or two small chromatin dots. A small vacuole is usually visible as we can see here in our picture, and multiple rings and RBCs are frequently seen. So this is a micrograph of the ring form of your Plasmodium falciparum, and you would notice the distinct cytoplasmic ring with your scanty cytoplasm. You can see here in the central portion of your red blood cells. Next is for the developing trophozoite. The Plasmodium falciparum developing trophozoite typically consists of one or two rings that each possess a heavy cytoplasmic ring, which is thicker than in the preceding ring form stage. So fine pigment granules may also be visible as seen in the picture here. And mature stages of this form are not routinely seen in the peripheral blood. And remember that mature stages of these trophozoites are only seen in severe infections. So the most prominent characteristic here of your developing trophozoite over Plasmodium falciparum is your heavy cytoplasmic ring. So again, the developing trophozoite of your Plasmodium falciparum under the microscope. So you would notice the heavy cytoplasmic ring, which is well stained here at the middle portion of your cell. So as with other Plasmodium species, it also has both immature and mature schizons. Both schizons are only found in the peripheral blood of patients with severe Plasmodium falciparum infection and not routinely seen in the peripheral blood. So just like our developing trophozoite, it can only be seen in severe infections of Plasmodium falciparum and not routinely seen in the peripheral blood. For the immature schizon, it is characterized by multiple chromatin bodies. So we can see here multiple chromatin bodies here surrounded by your cytoplasm with while the mature schizon typically contains 8 to 36 merozoites in a cluster arrangement. So usually the Plasmodium falciparum mature schizon contains 8 to 36 merozoites that is arranged in a cluster-like arrangement. So under the microscope here on the left side of the picture, or the left side picture here, is the immature schizont, and on the right side is the mature schizont. So notice on the right side that the merozoites that is arranged in a cluster arrangement. So lastly is the gametocytes of your Plasmodium falciparum. You have your micro and macro gametocytes. So because of this similarity in shape, it is difficult to distinguish the macrogametocytes from your microgametocytes of your Plasmodium falciparum. So the typical Plasmodium falciparum microgametocyte assumes a characteristic sausage or crescent shape, and as we can see here, there is the presence of your dispersed central chromatin and a nearby black chromatin. So usually the gametocytes of your Plasmodium falciparum are sausage shape or crescent shape. Lastly, the macrogametocyte also has a sausage-like or crescent-shaped appearance. However, it differs from your microgametocyte since its chromatin is usually more compact than the chromatin of the microgametocyte. So that is why it is labeled here as your compact chromatin. So the microscopic features of your gametocytes under the microscope, you can really see the sausage-like appearance of your gametocytes.
Moving on to Plasmodium knowlesi. Morphologically resembles to Plasmodium malariae to the extent that there is documented evidence that misdiagnosis by microscopic methods has occurred. depending on the morphologic forms present, may resemble to Plasmodium falciparum. Plasmodium knowlesi is a parasite that causes malaria in human and other primates. It is extremely rare, and because of the following number of reports of human knowlesi malaria infection detected by molecular methods, it has been recognized to be the fifth species of Plasmodium that infects humans. Now, as you can see, there is no detailed description of the morphology of the Plasmodium knowlesi, yet early trophozoites may resemble to Plasmodium falciparum, and other stages also resembles to Plasmodium malariae. Moving on, we are going to discuss the summary of parasite features, starting off with Plasmodium vivax. It has an enlarged erythrocyte size. Schnuffner stipplings are present in all stages except for the ring forms. Talking about the parasite cytoplasm, it is irregular, amoeboid in trophozoites, and has a spread out appearance. Now, the parasite pigment is golden brown and inconspicuous. The number of merozoites has an average of 16, and it is in all stages of circulating blood.
Now for the second parasite, Plasmodium ovale, it has an enlarged appearance similar to that with Plasmodium vivax. But what differentiates it from other parasites is because of its elongated erythrocyte. Schaffner stippling is present in all other stages but not with the early ring forms. James dots. The parasite cytoplasm is rounded with compact trophozoites, occasionally slightly amoeboid, and its growing trophozoites have large chromatin mass. Plasmodium ovale is dark brown in color and is conspicuous. The average number of merozoites present is only 8, and all stages can be found in the circulating blood. For Plasmodium malariae, it has a normal erythrocyte size appearance. Schaffner stippling, Siemens dots. We can determine that the parasite cytoplasm, the appearance of parasite pigment, the number of merozoites, and the stages found in the circulating blood is similar to that with Plasmodium ovale. It has a rounded, compact trophozoites, but with dense cytoplasm. It is also dark brown in color and is conspicuous. It has an average number of merozoites, which is 8, but rosette schizons, a microscopic feature, is occasionally seen. All stages are also present in the circulating blood. For Plasmodium falciparum, the appearance of erythrocyte size is normal, but it has multiple infected red blood cells which are common in this parasite. Schaffner stippling, Morris dots. The parasite cytoplasm has young rings which are small, delicate, and often with a double chromatin dot. Gametocytes are crescent or elongate. The appearance of parasite pigment is black in color, and it has a coarse and conspicuous in gametocytes. Plasmodium falciparum has an average number of merozoites of 20 to 24. Among the 5 parasites, it has the biggest number. However, the ring form and gametocytes are only found in the circulating blood. For Plasmodium knowlesi, there is no table because it is considered indistinguishable. So to review, among the 5 parasites, only the Plasmodium knowlesi is indistinguishable.
Laboratory diagnosis. For laboratory diagnosis, we have the PCR, flow cytometry, fluorescence microscopy, malarial blood smear, and malarial antibody and antigen detection. Polymerase chain reaction. The polymerase chain reaction is based on the principle of enzymatic replication of the nucleic acids. The PCR makes it possible to obtain by in vitro replication a multiple copies of a DNA fragment from an extract. Matrix DNA can be genomic DNA, complementary, or mitochondrial DNA, in which the complementary DNA is obtained by RT-PCR from a messenger RNA extract with the addition of poly-A-RNA. The amplification of DNA sample is based on the replication of a double-stranded DNA template. It is broken down into three phases: the denaturization phase, hybridization phase with primers, and an elongation phase. The exponential amplification is achieved since the products of each synthesis step serve as a template for the following steps. Polymerase chain reaction is an alternative method for malarial diagnosis. It allows sensitive and specific detection of Plasmodium species DNA from peripheral blood. Polymerase chain reaction may be more sensitive than conventional microscopy in very low parasitemias. It is also used for more specific Plasmodium species identification. However, it is not suitable for routine use. Flow cytometry. The basic principle of flow cytometry is the passage of cells in single file in front of a laser so they can be detected, counted, and sorted. Cell components are fluorescently labeled and then excited by a laser to emit light at varying wavelengths. There are several detectors that are carefully placed around the stream where the fluid passes through the light beam. One of these detectors is in line with the light beam and is used to measure forward scatter or FSC. Another detector is placed perpendicular to the stream and is used to measure side scatter or SSC. The analysis and detection of malaria infection by flow cytometry makes almost exclusive use of fluorescent dyes which are specific for nucleic acids. DNA-specific dyes are especially used because the parasites multiply inside the RBC or the red blood cell population of the blood cells. Since the RBC does not contain DNA, the DNA-specific fluorescence from infected RBC can only be due to fluorescence of dyes bound to parasite DNA. Parasites multiply within the RBC by several mitotic divisions. The fluorescence intensity of stained parasites increases during development of the parasites. The nucleated blood cells can easily be distinguished from parasites on the basis of the difference in fluorescence intensity. The fluorescence microscopy for malarial diagnosis is the use of a microscope fitted with a source of ultraviolet radiation to aid in the examination of fluorescent blood specimens to detect malaria parasites. It involves fluorescent labeling, in which is a process of covalently attaching a fluorophore to a target molecule, generally done by using a reactive derivative of the fluorophore that selectively binds to a functional group contained in the target molecule. A fluorophore is a fluorescent chemical compound that can re-emit light upon light excitation. The molecules to be labeled are antibodies, proteins, amino acids, and peptides, which are then used as specific probes for detection of a particular target.
Next is the malarial blood smear. So malaria blood smear, it is the gold standard for malaria diagnosis. It is stained with gemstone stain for parasitemia quantification, species differentiation, and better visualization of life cycle stages. So ideally, gemstone stain is used to quantitatively and qualitatively describe the Plasmodium species that may be present during microscopic examination. Other than that, right gem sustain can also be used for rapid results. However, cytoplasmic stipplings such as the Maurer dots, Schaffner's dots, Siemens dots, and James dots become hardly visible. So there are two types of blood films prepared in malarial blood smear. First is the thick blood film or smear, which is used for the screening of parasites. Next is the thin blood film or smear for species differentiation and identification. So in thick blood film, it allows more efficient detection of parasites since it is more concentrated, and it can also give the microscopist the rough estimate only of the degree of parasitemia. Thus, the thick smear is always examined first before the thin smear, but only in thin blood film where the microscopist can quantify the number of parasites. Thin blood film or smear. These are the factors to consider during thin blood film examination. First is the appearance of infected RBC, which can indicate the Plasmodium species and the stage of the parasite. Take note that RBCs can only be found in the thin smear because it is the only preparation that is fixed. The second is the parasite's appearance, and the third is the stages found. So these are the characteristics of a good preparation or an ideal thin blood film or smear preparation. So a dense body, this should take up about two-thirds of the entire smear, a monolayer area just behind the feathered edge, and a well-developed feathered edge. So prior to staining, if the slide is held up to the light, there is a rainbow effect seen. So the shape of an ideal smear resembles that of a short tongue. It should have margins on both sides. So for thin blood smear, the three layers that must be seen from the thickest layer to the thinnest layer are first the body, which is the thickest, second the monolayer, and third is the feathered edge. For the preparation of thick and thin blood smears, you can utilize capillary blood or EDTA collected venous blood. So in preparing thick blood smears, first place 3 small drops or 1 large drop of blood onto one end of a clean microscope slide. Second, using the corner of a clean microscope slide as a stirrer, combine the contents of the 3 drops of blood by thoroughly mixing and spreading to a circular film approximately the size of a dime or nickel. This is to prevent fibrine formation, which can obscure during microscopic examination. Third, let the slide air dry, and fourth, remove the hemoglobin by immersing the slide in a buffer solution before staining or directly during gem staining. So be careful not to fix the thick smear to allow the red blood cells to be dehemoglobinized. This is why only lysed RBCs are found in thick smears. So for the preparation of thin blood smears, first you need to hold the second slide or spreader slide on edge at a 30 to 40 degree angle and draw back into the blood drop, allowing it to spread along the edge of the spreader slide. Second, quickly and steadily push the spreader slide forward so that the blood spreads out into a thin film with a feathered edge. Third, you need to air dry and stain the thin blood smear. So again, only thin smears are fixed with methanol. So these are the steps how to stain the blood films using GemSac. So in thick smears, first you need to stain with GemSac stain with ratio 1 is to 50 dilution of GemSac stock solution with buffered water na 7 ang pH for 30 to 60 minutes. Second, you need to wash briefly by immersing the slides in the buffered water for 3 to 5 minutes and drain. Third, you need to air dry in a vertical position and examine under oil immersion objective. So in thin smears, first you need to immerse the prepared slides for 1 minute in a Coplin jar that contains absolute methyl alcohol or methanol. This is the fixative. Second, you need to let it air dry. Third, you need to stain it with Gemsastain with ratio 1 is to 20 dilution for 20 minutes. Fourth, you need to wash by dipping slides in and out of buffered water once or twice. And last, air dry in a vertical position and examine under oil. So, but you should always first use kaya ang LPO, then HPO, then OIO since microscopes nowadays are parfocal man. So, since the thin film was already um, fixed before washing it with buffered water, thus RBCs are still preserved yapon sa specimen.
Table 6.1 occur. Table 6.1 contains data on the occurrence of cyclic paroxysms in common Plasmodium species. For Plasmodium vivax, it is every 48 hours. For Plasmodium ovale, it is every 48 hours. For Plasmodium malariae, it is every 72 hours. And for Plasmodium falciparum, it is every 36 to 48 hours. It is important to note that blood specimens for malarial blood films are ideally collected just before the onset of fever or during the onset of paroxysm. Considerations for malarial blood smear collection and preparation. Prepare smears as soon as possible after capillary blood collection, and avoid fixing thick smears. For the examination and parasite quantification, please refer to the malarial smear lab discussion video that has been uploaded. Microscopic examination and findings. For Plasmodium vivax or Plasmodium ovale parasites, there are enlarged infected erythrocytes. Under this is Schaffner's stippling, which is usually seen in infected RBCs with Plasmodium vivax and Plasmodium ovale. However, it may not be visible in infected cells with early ring forms or slides stained in a not appropriate pH. For Plasmodium malariae and Plasmodium falciparum, there are normal-sized infected RBCs. Degree of parasitemia and clinical correlations. For 0.0001 to 0.0004% parasitemia with 5 to 20 parasites per microliter, the remark is sensitivity of thick blood film. For 0.002% parasitemia with 100 parasites per microliter, the remark is patients may have symptoms below this level where malaria is seasonal. For 0.2% parasitemia with 10,000 parasites per microliter, this is the level above which immunocompetent individuals show symptoms. For 2% parasitemia with 100,000 parasites per microliters, This is the maximum parasitemia of Plasmodium vivax and ovale. For 2 to 5% parasitemia with 100,000 to 250,000 parasites per microliter, it is hyperparasitemia or severe malaria. There is increased mortality. For 10% parasitemia with 500,000 parasites per microliter, there is exchange transfusion. For 10% parasitemia with 500,000 parasites per microliter, exchange transfusion may be considered. There is high mortality. For parasite density estimation or an alternate method, the number of asexual parasites per HPF on a thick blood film are examined. 1 plus or plus 1 to 10 parasites per 100 HPF. 2 plus or 2 plus symbols. 11 to 100 parasites per 100 HPF. 3 plus or 3 plus symbols. 1 to 10 parasites per each HPF. 4 plus or 4 plus symbols. There is greater than 10 parasites per each HPF.
Next is the quantitative Buffy code. The QPC is a fluorescence microscopy-based malaria diagnostic test. It also speeds and simplifies malaria detection with a combination of features and benefits. It has two principles, which is the fluorescent stain and centrifugation. The fluorescent stain is eternal coated with fluorescent acridine orange stain to improve the visibility of malaria parasites. When excited by blue light, parasites will fluoresce brightly against a dark background. Centrifugation. When the tube is centrifuged in the QVC capillary centrifuge, blood components and malaria parasites separate based on density and concentrate in distinct layers. The advantages and disadvantages of quantitative Buffy coat. Advantages: rapid and reliable in diagnosis of malaria even under field conditions. Requires less training and experience than blood smears. It is also as sensitive as thick smears. Disadvantages: since results cannot be used to speciate Plasmodium, a thin smear must be examined. Ring stages of P. falciparum and P. vivax are difficult to distinguish. This problem is particularly important in endemic areas where P. falciparum coexists with P. vivax. As you can see in the image, the tube is coated with acridine orange, then blood sample is collected in tube and centrifuge. Lastly, it is examined under fluorescent microscope. Layers are formed and positive result appears a green color. Malaria antibody detection. Malaria antibody detection for clinical diagnosis is performed using the indirect fluorescent antibody test. The procedure can be used as a diagnostic tool to determine if a patient has been infected with Plasmodium. Principle: the patient's serum is exposed to organisms. Homologous antibody, if present, attaches to the antigen, forming an antigen-antibody complex. Fluoresce in labeled. Anti-human antibody is then added, which attaches to the patient's malaria-specific antibodies. An apple green color indicates a positive reaction has occurred. The image shows a positive IFA result with P. malaria's schizone antigen. It is not routinely used for diagnosis, its transfusion blood screening, investigation of cryptic malaria, epidemiological purposes, and blood stage antigen is used. Lastly, in malaria antigen detection, various test kits are available to detect antigens derived from malaria parasites. Its principle: the immunochromatographic test device is coated with monoclonal antibodies. Visible lines are observed in the test region if specific antigens are present in the blood sample. The histidine-rich protein 2 is present only in P. falciparum. This test region will give positive result only if the sample contains P. falciparum. The lactate dehydrogenase is important for all Plasmodium species. This test region will give positive result if the sample is all Plasmodium species. If both test regions show positive result, it indicates either an infection with P. falciparum or mixed infection. In the image, number 1 is pure or mixed infection with P. falciparum. 2 is non-falciparum malaria, and 3 is a negative test result. 4 is positive with other Plasmodium species, and 5 it is invalid test result. Other diagnostic tests are intraleukocytic malaria pigment and lastly the mass spectrometry. And that's all for the Plasmodium species. Thank you.